This first compound is called nordinine, and this is a substituted, what we call a phenylethylamine. This benzene ring is the phenyl, the ethyl are the two carbons here, and then the amine is this N. In this case, it's a dimethylamine. So these compounds, phenylethylamines, tend to be, at least in the pharmaceutical sense when we use them, they tend to be psychoactive compounds. They're compounds that are active in the central nervous system. Frequently, some of these compounds cause hallucinations, but they have other beneficial effects as well. Anyway, this particular one, nordinine, is actually extracted from a red algae called Phylophora crispa, and it's a diuretic. That is something that will help with the elimination of water. That is, it will produce more urine. So when people get bloated, this is a way of eliminating some of that excess water. It also used to be used as an anti-diarrheal agent, but there are better compounds for that now. So it's no longer used for that. Below that is another, it's actually another form of phenylethylamine. It doesn't look like it, but let me see if I can hold this upright and show you this. Here is the phenyl ring. Here's carbon 1, carbon 2, that's the ethyl, and here's an amino group. But when this comes around, cyclizes around here to form a ring, so we've got a 6-membered ring and a 5-membered ring with a nitrogen in it. That particular ring is called an indole. And indoles also have the same kinds of psychotropic activity. Things like serotonin, which is a neurotransmitter, have an indole structure to them, and illicit pharmaceuticals like LSD also have an indole component to them as well. This particular one is called cholerpin. It's got an indole on one side, and it's got another indole on the other side. It's upside down from the perspective that we saw before. In the middle is a 8-membered ring, what we call a cyclooctadiene, because there's 2 double bonds in it. And that compound called cholerpin is isolated from a green algae called Caulerpa racemosa. It's an antibacterial and antifungal compound. The reason that you've got these antibacterial and antifungal compounds in the algae is because bacteria and fungi, these kinds of things, would actually try to grow on the surface of the algae, and this is one of the ways that the plant has to protect itself from these pests. It also is a plant growth regulant, and when it's used in this is actually can be used for agricultural purposes. So the other thing that it does is it inhibits the formation of a kind of plant tumor that's called a crown gall that actually forms on roots of certain kinds of plants. I've got another indole right underneath this. You can see this indole. This is called fragilimide. Kind of a big molecule here. Here's the again the indole rings right here, and then you've got a side chain off of that that's got an amide, a CONH, and then there's an amino group down here, and another phenol, which is a hydroxy benzene ring over here. And this is derived, the fragilimide is derived from Martensia fragilis, a red algae, a red macroalgae. This is a strong antioxidant. So you might recall that antioxidants are these kinds of materials that prevent, they actually scavenge things like free radicals, which are involved in accelerating the aging process and probably in certain kinds of disease conditions. So antioxidants are good things to have in the diet. Under fragilimide, I've got another compound, a much different compound, derived from sugars. This is called dihydrothursiferol, and dihydrothursiferol is a fairly complex molecule. Let's see if I can show you that right over here. So these 6-membered rings are called pyranes, and there's an oxygen and then 5 carbon atoms, and you can see there's more pyranes here. These are this is what makes up sugars. So these are derived from polysaccharides in the organism. And this particular one is isolated from Laurencia viridis, another red algae, and it's an anti-neoplastic, which means that it actually inhibits the growth of certain kinds of cancer cells. These happen to be breast cancer cells. So this is an interesting, has compounds like this. Rarely are these the things that ever wind up actually getting on the in the pharmaceutical market, but it's these kinds of things. These kinds of things will actually be used as a backbone, and then chemists will change the structure somewhat to try to potentiate the activity, to make it less toxic and make it more efficacious. So, but again, this is something that does show some potential. Underneath that, I've got a nitrogen heterocycle that this is called. This is a pyridinopyridine, and this particular compound is called lofocladine. And lofocladine, which is a fairly simple structure, this is an alkaloid. These are nitrogen-containing nitrogenous bases that are produced by plants, and in this case, this particular algae, which is called Lophocladia. And Lophocladia is just the genus. There are a number of species of Lophocladia, and all of them seem to produce this material. This is cytotoxic to two different kinds of cancer. One of them is designated CH06, and it's a type of lung cancer, and the other one is MDA-MB435, and these are breast cancer cell lines. So in both cases, this material seems to inhibit or kill the cancer cells. Below this is another compound that is derived from a sterol, and this is called stipoldione. The organism that it's taken from is Stipopodium zonale. It's a brown algae, and this is kind of interesting. Those of you that took the Jerusalem Science Contest last year, the one where we spent a lot of time talking about DNA, may remember a chemical compound called colchicine, which actually was a material that would inhibit microtube assembly, and it actually stopped mitosis dead in its tracks. So this is a potent mitotic inhibitor in mammals, and it also is very potent in sea urchin embryos. And there's probably a reason for that, because again, as I said, what the algae is trying to do is mount a form of chemical defense. It turns out that sea urchins feed on these things. So if the algae can kill the embryos, then that reduces the population of sea urchins and at least gives the algae a fighting chance. So as I say, this material also has, I didn't mention this, but it has anti-tumor activity against D388 lymphatic leukemia. It's very, very toxic to fish, but it has fairly low mammalian toxicity. So it may be toxic to fish, again, for the same reason that fish are also going to wind up eating this material. Then we have another compound. So that was the one that I just showed you, the sterol-derived compound. That was stipoldione. And then we have something called fucoidin, which is isolated from fucus. And this is the monomer of the fucoidin molecule. So fucoidin actually has, this is actually a polymeric structure. It turns out that the fucoidin is a polymer of this monosaccharide. This is a kind of simple sugar. And this is actually isolated from something called Laminaria japonica and other Laminaria species. This compound is cytotoxic. It will kill L1210 leukemia. That's been demonstrated only in mice, not in human beings. But I think it's still undergoing some kinds of trials in that area. It has a lot of other interesting pharmacological activity as well. It's also an anticoagulant, which is particularly interesting to me as I recently developed, as a consequence of my surgery, a thrombus or blood clot in one of the veins deep in the lower part of my leg, called the Solaris vein. And anyway, this particular compound, one of the things that was given to me to kind of prevent this clotting from taking place, it apparently didn't work, but it's a low molecular weight type of heparin material, which unfortunately, as I said, this didn't function. But it turns out that this particular compound, this fucoidin, is supposed to be about 2.5 times more effective as an anti-clotting agent than the heparins are. And heparins are actually used. It's a standard way of preventing clotting. When you give blood, for instance, the tubes, one of the tubes is usually heparinized to prevent the blood from clotting in the tube. So it's an anticoagulant. It also has antiviral activity, anti-neoplastic activity. I mentioned the leukemia, but there are other cell lines that it will actually. It's also effective against these other cell lines. It has anti-inflammatory activity, immunomodulatory activity, and it's an antioxidant. So this particular molecule seems to have a lot of potential biological uses. There's another one down here that I want to show. It's kind of interesting molecule. You'll notice that all of these have very diverse kinds of structures. What's interesting about this is this 3-membered ring, this little triangle here. That's called a cyclopropane. And we don't see that so much in natural products. It's a very, very strained ring. There's a lot of energy that goes into making a ring like that. But nevertheless, this is a compound. This one doesn't have a simple name like the other ones do, but it's isolated from Halimeda species, and this compound is antibacterial and antifungal.